Assalamu alaikum. I hope all of you will be fine with the best of your health. So today I am going to explain another topic from the NCBI series. If someone is there who has not taken my previous lecture of the NCBI series, he must have first go and watch it, because I'm not going to explain the previous concepts in this lecture. Maybe I'll touch some of them, but I'll not explain completely the previous lecture, like the gene section of the NCBI series. Basically, we have different resources and different databases which are merged in the NCBI, and I have already explained one of these databases named Gene or GenPept database. I have completely explained and discussed all the important concepts about this database in my previous lecture. And now in this lecture, I am going to explain the next topic, which is the protein information or the protein database. The name of the database in this NCBI is basically GenPept. So let's get started. First, I will choose the database name, that is protein, from the list, as you can see. Then I'll write here the name of the protein, that is tox. As you can see here, different hits. I've got different hits when I have searched the name of the gene tox. But I can specify the name of the organism also here in the query box. I can write here the name of the organism or the specific organism in which I want to find the protein, or I want to find this protein named tox. But I don't want the gene information or the GenPept information which I have got at the top of the hits. So I'll ignore it because I have already explained it in my previous lecture. Here I have got the protein information. I'll go for the protein information or the hits which are explaining about the protein information, which means that this tox gene is producing or converting in a protein named tox. Like, as we all know that a gene is converted into the protein, and ultimately it will perform its function by converting into the protein. Actually, that protein will perform some specific function. So as you can see here, I have got the 116 amino acid protein. AA basically means amino acid. So which means it clearly denotes that this is the protein information. And I'll just click on this option. But before that, let's see these different hits. Now I'm going to write the name of the organism in the query box right after the name of the protein tox. Let's see what happened or what we returned at. Because when we specify the name of the organism right after the gene or the protein, it will search in that specific organism, which will help us ultimately in finding the correct gene in the correct organism. In its name from Homo sapiens to mus musculus. So now here you can see that we have got this gene at the top of the hit, that is tox, in that specific organism named mus musculus. Actually, this thing will help us in finding the gene right in the organism in which we want to find this gene. So I think it's my suggestion that you have to mention the name of the organism also along with the name of the gene. In this case, you don't have to, go into the nitty-gritties of these hits. In this case, you don't have to mess up all these hits or search and the direct result in that specific organism for that specific gene. So yeah, my suggestion is that you have to write the name of the organism also in the query box along with the name of the gene. This will give you a direct result and will also save your time. So let's just see the results of this query. From here you can see, and this is the name of the organism, and right after that you can see a ID after the topic, that is GenPept, and after that you can see the name of the ID. This is basically a GenPept ID. As I have already discussed in my previous lecture all details about the GenPept, so go find what is GenPept ID from here. This is basically the gene ID or the GenPept ID. Actually, these IDs are most of the time repeated in different databases so that we can keep track of the things, that either we are going at the right gene or in the right protein, or we are searching the right gene which is converting into the right protein. Like sometimes people search some gene and they search another protein which is not converted from that specific gene, and they just mess up all these things and make things wrong. This database is basically the GenPept database, and this is a protein database, or all the information which is present in this database is related to protein. This is the GenBank ID, which is a gene information ID, not the protein information ID. But this ID will denote or confirm us that we are searching in the correct protein which we have previously attained. This ID is basically very important because this ID will lead us to the page related to the same gene information of this same gene in the same specific organism. Like in this way, we could not mess up all these things. We will keep track and go in the right direction. So, as you know, locus is the location of the gene, and in the next box, we have got the ID for that location, and then we got the length of the amino acid, that is 320 amino acids, and then we have got the structure of the protein, that is linear. As we know that protein has different level of structures, and linear is the very basic structure of the protein. And then we have the next option, next thing is ROD. ROD is basically reviewed organism diverse, which means it is basically the date at which indicates that this sequence or the sequence of the protein has been reviewed in that specific organism, and this sequence is basically from the set of diverse protein sequences, right? So this is a basically important date which is important for most of the time for the researchers. And then we have the definition, and then the accession number. Accession number is the most important thing of this database, because as we all humans have our identity card, accession number is basically the identity card of a protein or a gene. So yeah, this is very important for them. Without them, they don't have any identity. Then we have the variant. The variant is basically has the information about the updation of the information. Like different researchers do research and update according to their research. So as their research is updated, the variants are also updated. The next thing we have is the source. Source is basically the name of the organism, that from which organism this protein has been extracted. And then next we have the organism, which tells us about the lineage of this organism, like the complete lineage. And then we have the most important thing of this database, that is reference, because without this all the information is totally fake. Because reference is a thing which tells us that this information which we are going to study or which we are going to have in our further research is basically authenticated, proven, and published. So yeah, this is the most important part of this database, because without reference you cannot consider any of the information authentic. What is this what we have got in this reference? Basically, in the reference we got the article name. What happens here? Basically, some group of researchers or scientists do research on specific topic or that specific protein, and then they prove or discover something new, and then they make a report of this thing which they have discovered, and this report is basically known as article. So here we have got the name of the article in the title section, and in the authors we have the name of the scientists or the researchers which have studied at this specific protein and proved their results, and their results have been approved and published in the world. And by this research, they have proved in the world that they have discovered this thing, and this thing is very authenticated because a team of experts basically approve their research article. And they found, if they found any type of small or tiny ambiguity, they never accept and never publish the article. So yeah, this is the complete science of a research article basically. So as you can see, the name of the article is a novel PEPP box gene tox is highly glutamic acid rich and specifically expressed in murine, testis and ovary. And here we have the name of the journal. Name of the journal is basically the company which approved their research or their article and published their article in the world. So the name of the journal is here Biology Reproductive. And basically this is the abbreviation of this journal. The complete name of this journal is Biology of Reproduction. And next is the name of the volume, that is 70, and after that we have got the page number 828 to 836, and next we have the date or of year, publishing year, that in which year they have published their article. So in this way, and then next we have the article link. Basically this PubMed is the company which provides the link for this article. We can search or click on this link. I am going to open it in the next tab. As you can see in the next tab, this complete article is going to open. So as you can see here, this is the complete article which is opened here. This article basically about that specific tox protein, and here you can see the name of the researchers or the scientists, and this is the PMID, which means the PubMed ID, and the DOI basically states that digital object identifier, that you can directly open this article by just clicking on this its DOI link. Every research article has its DOI link. And next you can see the abstract of this article, and after that you have other similar articles which are related to this article. If someone is interested in more articles which are also related, you can see there, but I am not going to explain and explore the next because I want to connect my focus on the same place here. So this is the second reference. Just like before, we have there in the second reference we have another article and another group of researchers or scientists. Here you can see the name of the scientists, and then the journal name, and then the date of publication there. And similarly, just like I have already explained the reference number 1, this is the reference number 2, another article which is proving this page or this database, that this information which we are going to get in this database is really correct and authenticated and proven. So let's explore the features here. This is the information which I have already told you, like the organism is mus musculus, and chromosome is the basically location of this gene is at chromosome X, and the tissues from where this gene is extracted is testis and ovary, and then product is basically the gene is the input and protein is the product or output of this gene. So the name of the product means that we have the protein information of this tox gene. So here we have the chromosome information, and then the source from where this gene has been extracted, right? And similarly 1 to 320 is basically the amino acid number, and then the product as I have already discussed it, and then we have the name of this protein, homeoprotein, right? So this is the some of the basic information of this protein which is related to this protein, and then the region which is specific for this protein, that is homeoprotein, which is part of this protein, and then we got the next information like Note PFAM is CDD. PFAM and CDD are the databases which most of the time, basically these are the databases which tell us about the domain of the protein. Like if whenever we see in the database the name PFAM and CDD, these are basically the specific domain databases which are just specific and specialized only for finding the domain of the proteins. So here we have got the domain information also, uh, not just the protein but also the domain of the protein also we can get from there. Here is the link, we can directly click on them and go at this domain information of this protein. As we all know that all part of gene is not coding, some part of the gene is coding because we have ORFs, open reading frames, in our gene sequences. So therefore we have some parts of proteins which are genes which are going to be converted into the protein. So here we have these parts of the protein, and there we got all the protein sequence which is completely converted from the gene. And this is, there we got the complete details of this database. So this is the end section. So now this is it for now. And yeah, here you can see the blast section also. You can run directly from here the blast also if you want to blast it, but I have not covered the blast section. I will also cover the blast section in my coming video. So from here you can get the complete full text also. As you can see, this arrow, small arrow, if we click on them, we can get different other options for downloading or changing the format. Like if we want to get the faster sequence, we can get the faster sequence from here also, and we can download this sequence or copy this sequence and paste in a notepad file just by clicking, just by selecting it and copying it and then pasting it into a notepad file. This is the GenBank ID as and the accession number. So this is the accession number and this is the name of the protein and organism name. Here in this option, identical protein, we can get the identical proteins. And in this section we have covered almost all the information about the protein. So as the GenBank ID is also there, if I write directly this ID in the query box and select the gene database from here, then we can get also the gene information directly from there. Like if we have the ID, we can just get this information. We can go directly at this page, right? So it's just easy. Like you can see, this is the gene ID here. It means that you are going to explore all the gene information here. So if you want to download directly this file or faster file, then you can create this file by just choosing these options from here. So you can see I have downloaded this file and it's automatically created and downloaded. So here I have got the faster format of this protein sequence. So yeah, that is it. This is so easy. There is no difficult science in here if we keep things like in track and make things right in the right direction, in the right way, then yes, we can do this research. And this is my personal experience that I feel so easy and satisfied and comfortable by these databases, because once you have become the expert of these databases, these become very easy for you. So thank you so much for that, for watching my video. If you have not subscribed my channel, then just subscribe my channel for upcoming new other videos. Thank you so much. Thank you so much. Thank you so much.